start my recording. Hopefully everything will be okay. And oh yeah, I have to share my screen. Okay. Does everybody see my screen?
It's just loading for me now. Yeah, I can see it.
Great. I was just going through my slides. So the way I structured the slides actually is more of a, it's a presentation, but it's more of a notes that I took from some paragraphs from the paper. And yeah, they're kind of structured already the way the paper is structured in a way. Some of them are quite text heavy, but I think that's all right. Usually don't like text heavy slides, but yeah. I mean, okay, yeah, if we're starting, then today we're looking at automated search for artificial life. It's a paper. Actually, let me put it in the chat if we don't have it. So this is what we're looking at today. They have a like really nice, it's not interactive, but they have lots of GIFs and videos here in the article. In the paper, okay, let's go through the slides. So what they do is they give us this automated meta algorithm, let's say, that can search through the space of a simulation, and they look at 3 different types of search that you can do. So one they call supervised. You just have you run your simulation and you supervise it to look as a, you give it a prompt, and then the simulation kind of is supervised to look the way that the prompt is set up, and they do this alignment using clip. They actually use clip for everything visual, and here also textual. The other type of search they do is open-endedness, and open-endedness they define as the ability of the system to produce diverse outputs continuously. So this is kind of like diversity optimization here. And for the last type of search they do is, so you have a simulation which is parameterized with some parameters, and you kind of want to find different, very diverse and different types of simulations and kind of show them one next to each other. It's kind of similar as the one above, but also different. We can see it in the diagram they have. They, for some reason, call it illumination. I'm not sure why. This the last type of search that they present. And what are the types of simulations that they apply this to? So they use neural cellular automatons. I would assume everyone's kind of already familiar with NCAs. They also use voids. And if anyone is have any questions, you can just stop me and ask me for clarification, I guess, if I'm aware. But yeah, voids are like, so you have these entities into this space, they can move, and they are aware of their surroundings, and they have a function that, depending on their surroundings and the other entities around them, they can change their behavior and direction, and they move like flocks. They usually simulate birds or fish. We have particle life, where we have particles, and based on their distance and some function they have, they can interact with each other. And also we have classical Conway-like cellular automatas. What are their major results? So they find previously unseen lenient Boyd's life forms, very open-ended Conway's game of life like CA rules. So they use foundation models, and they use the foundation models to quantify the emergence in those simulations. And okay, yeah. So okay, so these first few slides are kind of different anecdotes I found interesting from the article, just in the abstract and the introduction. So they're kind of a little bit chaotic, but yeah. Anyway, so yeah, what they say is the most important aspect of artificial world is the rule of the world, and they propose a new way to search by describing the phenomena that you want to use in natural language, and through the use of foundation models you can guide that search. Yeah, just describe your phenomenon and let the automated search like find the parameters of the simulation that lead to that behavior. Related work, so here's kind of a big wall of text of some things that were in that section of theirs, but yeah, so substrate is what they call the simulations themselves, so that would be the Boyds, NCAs, the CA, and the other one. So substrate agnostic, their algorithm only depends on the simulation has to be able to produce images, and if that's the case they can guide the search. Other related work, like they give like biomaker CA, NCAs with NCAs and linear. Like in linear there are different articles which optimize for specific behavior as well, but it's kind of limited to linear because you're optimizing for like speed. They also they just mention different algorithms like novelty search, but I guess it requires a good representation to be effective. Intrinsically motivated search. This is like you embed the different states of your simulation, and you kind of want them to be very different from each other, and they kind of do something similar, but they use compressors. They use foundation models to compress the space and get these representations that are aligned with like the real world in a way. Okay, yeah, human aligned representations. Other thing they talked about in this section was how they characterizations of emergence. They talked about like Kolmogorov complexity a little bit. So what we also have is assemble theory, which quantifies evolution by measuring the minimum number of steps required to assemble an artifact from atomic building blocks. They mentioned like computational reducibility, which is something that Wolfram, I think, is famous for. And one thing I like is their definition of open-endedness that they give. So open-endedness is the ability of a system to keep gathering interesting artifacts forever. But also may depend on the representation space itself, and also another nice thing that they said is like, while all interesting things are novel, not all novel things are inherently interesting. And so they use CLIP as a foundation model in this work, and they start to describe their procedure, their method, by first giving a few definitions of things. So what is a substrate? Those are the simulations. It's so the substrate is defined by some initial state, which is sampled from a distribution, let's say, and then an update rule, which you can call it on the state and get the next state, and then they also have like a rendering function, which you apply at the end, which gives you the image itself. And what you need from the foundation model, so the abstraction of the foundation model is just, you have to have a function which you can give it an image, and it will give you a vector representing that image, so like clip embeddings. And also, in order to be able to guide the search with text, you also need that for text. So again, clip, you can give it piece of text and it will embed it, and actually the image and the text embeddings are aligned. And this is also this is the dot product, which is what they use for similarity measure. And they after that give this nice overview of, like, nice picture of there are 3 different search algorithms that they describe. So in the next few slides we'll kind of look into them step by step, but you can kind of see. So the first supervised training, you have your simulation here, which is a sequence of frames. Every frame you pass it to the visual language model, and it gives you a representation. So each dot here is like a representation. This is like the clip embedding of every frame. And if you have a cell, 2 cells ecosystem here, for different time steps of the simulation, we have different targets which are kind of evolving. We're starting from a cell, then 2 cells, then an ecosystem. You can imagine they're like pulling each of those representations towards the representation of the text, which will kind of make it visually similar to the simulations, to what the text describes. Here I actually couldn't really understand specifically what they do here, because you have again a trajectory of the simulation in clip space, and for open-endedness you kind of want to have like a very diverse path. And what they do is for a sample number of points, and for like very close points to every point. So for this point is really close, and it kind of force this point to be further away from it. So in this way they are kind of making the simulation itself like walk in the embedding space like a bigger path, and more like winding path, let's say, I think. Yeah. And in the illumination here type of search they describe, here we have trajectories, and here we have the last frame of every trajectory, and we want to find a diverse set of trajectories. So what we do is we embed again all the trajectories, we get the last frame, and we're pushing away each of those trajectories. So kind of we want the last step of each simulation to be very different. We want them to be very different in clip space. Any questions so far? I would assume no. Okay. So yeah, it's kind of, it's not that actually complicated, I think, what they're describing here. It's very straightforward in a way even. But yeah, they also like explain that with formulas. I'm not sure if we kind of want to go deep in those, but again, yeah, you want to search. This is the argument. You want to find a simulation. The simulation is parameterized by what you find here that produces an image. So this RST function, if you remember from few slides ago, is the render function. So you run the simulation T steps, and then you render it with these parameters, and then you get the last frame here, and you embed it with the visual language model, and then again you embed the text prompt. This is your target, and you want them to be very similar. To be very similar, which would mean you want to maximize their cosine similarity, which is the dot product here. And so for the open-endedness, they do something similar, but they're doing an argmin over that space. They have, let's say, 2 different steps, T prime and T, and we want to minimize the cosine similarity, which would mean that they are pushed further apart from each other. And for the illumination, you kind of do the same thing, but so you get actually 2 different simulations. So here the theta parameter is actually what describes the rule of the simulation, and you have 2 different simulations. You get like the last step of each of them, and again minimize the similarity, making them very different. Yeah, and then you obtain a set of parameters which are actually a set of diverse simulations. Yeah, and then next use what I kind of like put them overlaid with this diagram they have, but I think it's kind of straightforward what they do, so I don't think we need to really go look at them again. So in their experiments, they use clip for visual and text embeddings. They also do some experiments with Dino. Dino has only can only embed images, so they use it only for these two types of searches that they do, the open-endedness and the elimination one, because yeah. And yeah, again, the substrates are voids, which their parameters, so all the births, let's say in the simulation, are parameterized by a neural network. And particle life, we have these matrices that kind of tell the particles how they interact with each other, and like some parameters that I hear Discord. Is there a question or something? Or simulation is focused extremely on search space exploration, trying to find different results from each other. Yes. Illumination is trying to find, again from this picture, so each of those lines is trajectories. We get the last frame and we're pushing them to be further apart from each other and searching for very differently looking trajectories. Yes. Don't see any other questions. Okay, so life like cellular automata. Yeah, this is like Conway's game of life. Is basically just you can imagine a Boolean discrete cellular automata, and so every cell counts the number of living cells around it, and based on that number it chooses whether it's dead or alive, and you have some parameters that vary that function. And I guess there are this many possible simulations, and they kind of use brute force just to search the space. For linear, I guess you have that many dimensions for the initial state and 5 dimensions for the dynamics. So these are the parameters of the linear model. And NCAs, yeah, they again are parameterized by a neural network. Yeah. Okay. And yes, here are some pictures from what they obtain. So this is the supervised training. This is just a single label, and we can see a multi-cell organism, a neural network of neurons, a virus, a caterpillar kind of produces something like that, a pepperoni pizza. And for voids, again, they're not that diverse voids, but they can definitely very specific also. Like if you give it like a Fibonacci spiral in nature, it kind of does this circular pattern. It's a starfish, a snake, a tree with roots. Yes, and for particle life, again, caterpillars look like caterpillars. A red organism, cell division, I guess. We can also look at these here, because in the article, in the HTML article, they actually have them as GIFs, which is nice to look at them like that. So neural networks of neurons are kind of chaotically sending signals through the different cell-looking entities. But yeah, they kind of look as the target of targets they're trying to approach. But they also have failure cases, and they showcase some of them. So they do single targets. So this is just give it a single label, and you supervised on the last frame. And for single targets, they run it on linear, voids, and particle life, and they use this algorithm, SEPCMAES, which is evolutionary strategies algorithm. So for evolutionary strategies, you don't need to pass and optimize with gradients, and I guess it will be harder. I'm not sure how particle life exactly is run, but it will be harder to run gradients there. For the oh yeah, and also they give this more examples here. Some of them we've seen, but for example, if you give like, so here we have artificial brain, a red organism and a green organism, and it kind of look as the label they're trying to approach. But I mean, if you give like a Boyd simulation, a green organism or a red organism, it's just not capable to give you something interesting because of how constrained I think they because of its lack of expressivity in that way. Okay, temporal targets. This is you give the simulation a sequence of targets to approach. They run it only on the NCA substrate, neurocellular automata. They optimize it with back propagation, atom optimizer, all the good stuff. Yeah, and if you tell it like one cell and two cells further apart from that, they say that the update rule, it learns to like enables like self-replication. And here we have some examples of that. Can I close this actually in this full screen? Oh, yep. So this is the example they give. So one biological cell, and they optimize it here, and then on further part in the simulation they optimize for two biological cells, and it kind of grows and produces this thing. And here we have more or less the same self-replicating molecules, cells floating in primordial soup, predator-prey dynamics, and alien animals. If you zoom in, they kind of look like aliens, the things from the from the last step here. Okay. Search for open. Oh yeah, that's the second thing they do for searching for open-endedness, and they kind of only run that on the lifelike CA, so the discrete simulation. So like Conway's game of life, but with parameters, let's say, and optimize the open-ended score. And because the search space is very small, they say they just brute force it. Not sure if they run like optimizations on like all of the simulations in a way, but yeah, I guess it's not that guided in this specific case. It's just, yeah, it's guided by the closeness of the representations they get. Okay, so here at the top we have Conway's game of life. If you optimize open-endedness in pixel space, you kind of get chaos, because this is the most diverse thing you can get in pixel space. But if you optimize it in foundation model space, so in clip space, you get very different things, which are different simulations for every run, which are also diverse temporally in a way. And they analyze it very interestingly here. So what we see here is, I think, this thing here, the blue one, is Conway's game of life. Here is the simulations they get when they optimize for open-endedness in pixel space. And here is what they get if they optimize for open-endedness in clip space. And these are, I guess, some dimensionality deduction algorithm that's run on clip space, or just they're taking a slice of the embeddings from clip. And what they notice is they get much wider and varying trajectories. So similar, they even get more diverse and winding trajectories than Conway's Game of Life. And another cool thing that they do is, okay, so oh yeah, okay, so what are they embedding? They're embedding the clip embeddings. They use UMAP as the dimensionality reduction technique, and they're able to map all of the space here in this plot, and they're coloring every dot is a simulation, and they're coloring it based on their open-endedness score. So how, I guess, far apart, so you get the trajectory, and for every point you get some points around it and measure how far apart they are, so how winding the path is, and you color it. And what they find is that the most diverse sets of trajectories live in separate islands that are further away from that main island, where, like, if you randomly sample, you'll probably get something from like this big island here. Yep. Yeah, interesting things they found is, so Conway's Game of Life, based on their open-ended score, is like among the 5 most open-ended simulations, the top 5% most open-ended CAs that lie on the edge of chaos. Is they neither plateau or explode? Yeah. And because the foundation's model of representations are related to human representations, producing novelty in the trajectory through the foundation model's representation space yields a sequence of novelty to human observers. Yeah, because they use like models like CLIP, which are trained on real things from the real world. What we optimize for novelty in that representation space, it is something that looks novel to us, I guess. And yeah, here are some more examples that they show at the appendix by running this optimization, which look interesting. Yeah, and so eliminating it. Did someone say something? Or no? Yeah, okay, nothing new here. So that's the last thing they do. They try to obtain a set of very diverse simulations. They find a set of very diverse parameters of those simulations, and like initial states. I keep hearing notifications from Slack. Oh, okay. It's okay. Okay. And so yeah, okay, they run these experiments only for linear and Boids. They use custom genetic algorithms to perform the search. So I guess at each iteration they randomly select parents, create mutated children, and keep the most diverse subset of the solutions. And diversity again is measured by the closeness in clip space. Yeah, so I guess in linear the algorithm discovers many unseen life forms, and in Boids very interesting new flocking behaviors, and such as snaking, I guess, and grouping and circling. I guess we can see it in the next few slides. So they also do this nice simulation atlas plot. They do it by so they run the search, they obtain a set of simulations. In each simulation they embed the last frame in clip space. They run UMAP and they embed it in 2D, and they place them right next to each other. So this is based on the how they kind of group them together on how they look, and it gives you that simulation atlas of linear. And here is what you get if you just randomly sample the search space of linear. Okay, they have a section of discussions at the end. So what they say is like the choice of the foundation models doesn't seem to be very to matter, because they run experiments with Dino again and kind of get the very similar things. But you have to run these metrics in foundation model space, because if you don't run it, if you run it in pixel space, you get like things you're not interested in. Okay, I think here on the last point we can just see the plot from the next slide. But yeah, they run some other like interesting experiments, like so if you interpolate between the parameters of one type of simulations, here we have something like cells, and here we have like this spiral thing that they were optimizing for, and if you interpolate between it, you'd see something in the middle that doesn't really make sense. It's not interpolating not sensibly, but yeah, it just shows the nonlinearity in the parameter space of Boyd's. More is different in particle life. So in particle life, where we have like points in 2D, and we're optimizing to find the right interactions between them so they can self-organize in something interesting. So for certain optimizations, you kind of need at least a specific critical number of points in order for you to achieve an interesting result. Here they say that you need at least 10,000 points in order to get like decent looking caterpillars. Yeah, why foundation models matter again. So if you run the search of Lenya and Boyd's in clip space or in Dino space, you get interesting diverse results. But if you run it in pixel space, you kind of get very chaotic and almost like normal noise looking patterns. Yeah, and this is my last frame. Questions and criticism from me. So they run open-endedness search only on the discrete cellular automata, and I'm not sure why they haven't run any experiments on Lenya and NCAs with gradient descent. Because they didn't specify a reason, they just said that they run the experiments only on the discrete cellular automata with brute force search. I think that would be an interesting thing to be seen. No temporal constraints. I mean, they do have temporal constraints in a way, because you can supervise to have specific behavior on different parts, and this is kind of temporally constraining the simulations. But actually they already mentioned it at the end that what they would like to have is, instead of, so here their constraints, let's go back to this diagram. Their constraints, they are independently embedding every frame of the simulation into clip space, and then they're doing the optimizations in clip space. And they mentioned that what would be interesting to try is to use a video model where you would be able to, I guess, embed either the whole simulation or like fragments of the simulation, where a single representation contains the information of a few frames and kind of contains the information of the dynamics itself in that way. Because this trajectory, these are like static points that represent the images in that space. They do not embed in a way the dynamics of the simulation. And that's basically it. You have any questions? Yeah, actually we can probably go through the article, or not the whole article here, but like just the GIFs they have, because they're nice. They're higher quality. Here again, the open-endedness search that they do only on the lifelike cellular automata. Did I see a question from Discord? No, Piotr is just writing. Okay.
I was wondering, Ilya, I wonder if the reason why they didn't run the open-endedness search on linear is if the space was possibly too big. Because if I remember rightly, they brute force. Did they search through all possible lifelike cellular automata?
Yes,
or not? I can't remember.
but the way I see it, you can optimize. So what they do is here they are pulling each of those points closer to the representation of a specific sequence of words, and they do that with NCAs using gradient descent. Why can't you, in this space again, run gradient descent and push these representations further apart? Maybe you're gonna get out of the, I don't know, this. No, actually, yeah. Why not? I think you should be able to back propagate through the role itself, right? Because the simulation is differentiable, the embedding is differentiable. So here in this space, you have a vector that's continuous function of like the whole simulation itself and the parameters of the simulation. I don't know if they have. Yeah. What do
I
you
just
think?
realized they actually use linear as the example of open-endedness, don't they? In that figure, they use
Yeah, they
linear
use
simulation.
they use linear here on the supervised targets.
And open-endedness, right? That's also a linear creature.
Yes, interestingly, they use it here, but they never use it actually in their experiments here. So their experiments on open-endedness are only on the lifelike cellular automata. So it's kind of misleading what they show us in the diagram. Yeah. Actually, yeah, I didn't notice that, but here, yeah, here again you see linear. Oh, yeah, I think actually they actually use linear for the last experiment. Yeah. So the in illuminating the substrates here in the atlas, they actually also use linear, and it looks really nice when you look at it like running like that here. Also the Boyd's one, if you sample them at random, you get these noisy looking ones which do not have anything interesting, and here they're kind of very more different
I'm sorry
from
if I
each
missed.
other.
Did you talk about how they modified the Boyd simulation? Because I remember that they mentioned in the paper that it's not a traditional Boyd's algorithm. There's some sort of neural network use for the sensing.
Yeah. So for the Boyds, so each bird or entity in the Boyd simulation is parameterized. Its behavior is parameterized with a neural network. So you can imagine a neural network that senses like the n closest birds, and depending on and their properties get fed into the neural network, and this outputs a direction, I guess, for every bird. And so it's a neural network, and what they do is, let me go back to no. Yeah, so for the illumination, so they use genetic algorithms to perform the search. They use linear and Boyd's, and I guess so at each generation they select parents and they mutate the children. I guess they do some sort of crossover between the networks, the neural networks, which are the things that parameterize both of these. I mean, this parameterized with the neural network, and this is just with a vector of parameters. But you can basically use genetic algorithms to search for both of these spaces. So
cool, thank you.
Yeah.
To clarify.
But yeah, it's cool article. Another criticism from me is they kind of say that this can give you very diverse things, but it's also constrained, the way I see it, to find things that you describe that are kind of seen in the real world, because clip space is learned from data sets from real life images, and you're also kind of constraining it to find, I guess, patterns that have already been seen. I mean, this could be like a desired behavior because this constrains the space a lot, but yeah, I don't know, it doesn't sound, I mean, the purpose is not to be generic in a way, but yeah.
I think I know what you mean. Like, I think in this case, in this context, it's actually a bonus that they're bringing in extra information because they're almost sort of replacing a human observer, right? They're trying to bring in
Yeah,
sort
but
of complicated, the
it's
ability to look in complex patterns. But it does raise the question: what would an ideal searching foundation model look like? Like an embedding that produces, like Clip or Dino, that produces embeddings that would be best fit for searching for diverse solutions. And what does diverse mean in this
I'm
case?
I'm not sure if the framework itself would be the right thing you'd wanna, this specific framework, because I don't I really like the, I haven't read the article
Sorry, one second. Apologies.
about intrinsically motivated discovery, but from the surface level understanding I have of it, I think I much rather like the idea of how they do the search. It sounds more generic in a way to me, I think, yeah. And yeah.
Sorry, I had to disappear.
It's okay. But yeah, it's kind of not very complicated what they describe here as a procedure. And oh yeah, also I run the notebook and we have some results, but I actually haven't changed it a lot, and so I guess we're not gonna get anything that we haven't seen. But yeah, I mean, if you supervise it here, you can. I have a nice notebook with lots of plots, and you can like change the substrate easily, and you can prompt it with something interesting. One thing I notice is like they I mean they're very constrained from the models they're working with, but and maybe that's the reason, because they have mostly used targets specifically for that search, which are kind of they're not very diverse, the targets themselves. So it's always like an organism or a cell or a neural network of neurons. I mean, I guess the caterpillars is kind of, the caterpillar one is kind of interesting, but and the pepperoni pizza, of course.
What would you like to see instead, or as well as what sort of prompts do you think would be most useful?
I the results they obtain are very diverse, but I don't know. Maybe they're not like that much surprising, and maybe I guess it's not much you can do in those spaces to get very different things. Actually, I think in NCA space you can get very different things from what you obtained from here. Very much, a lot more diversity in NCA space, but the only results we have are like this one, and they're kind of, I mean, they're interesting. I think they're very different from what we have here. I'm not sure why don't they produce. Oh no, it's actually a GIF here. I haven't noticed. It's not like a sequence, but it's actually a GIF. Yeah, I mean, these are nice results. I like the NCA results, I guess. I think they have the capacity to produce a lot more diverse things than the other things, but that's mainly because of the other models are a lot more constrained. I think they might have a little more pictures from NC experiments at the end here, which we haven't seen. Yeah. A starfish.
I think there's linear, aren't they?
Oh yeah, this is linear, sorry. So yeah, they targets on linear, targets on voids, supervised targets in particle life, open-endedness. They don't have, yeah, they don't have lots of examples. Actually, I think from what I've seen, that's the maybe the only example they have of NCAs. And
I wonder if they struggle with stability, perhaps,
yeah,
trying to
yeah,
get
maybe.
into an image.
But I think the NCA has would be the most promising to give you like actually very diverse things, because I mean they're very diverse, the results they have here, but all of them look like I mean you see it and you know that this is linear.
Yes,
in a
right.
way. And I think NCAs are very complex enough that they can actually produce things that you couldn't actually just by looking at it say that, oh, this is this. I mean, I guess you can guess that it's an NCA, but they could be very different from each other. I guess is what I'm trying to say. A lot more different than I guess linear.
I wonder if the NCA sort of tailoring an NCA, searching for one, would benefit more from having that thing you suggested earlier, which was the embedding that works over an entire video, rather than just being a single frame. But that said,
Yeah.
no, can you backpropagate throughout the entire rollout, like different, as in different rewards for different steps of a rollout? Is it possible to properly backpropagate that back through the model?
I don't see why
I'm
not.
sure.
I mean, you say that if you supply a gradient here, and a gradient here, can you backpropagate all of these at once? Is this your question?
Effectively, yes. Yeah.
I think yes, because I think gradients are like accumulating, and I think you can also do them in a sequence, and I think this would be good enough to guide you in the space. Like do a
That's
rollout
interesting.
and supervise like here, then do a rollout and supervise on the next few steps. But I think you can also do that at once, I think.
Okay, that's pretty cool. I didn't know that.
Yeah, because when I have run experiments with NCAs, I always actually supervised on the last like 5 frames at once. I don't like take only the last frame,
really?
only a single frame. What did you say?
That's cool.
Yeah.
I never heard of someone do that before. That's pretty cool.
Yeah, I mean, usually my target is static for all of the frames, but I don't see a reason why your targets cannot be different in different time steps. And actually, no, you know what? I had a simulation that, so I think I'm plugging myself every time that we have a discussion here, but what was it? What do I wanted to say? So for this is the I think I showed it to you last time for the classifying NCA. But it's not actually. Oh no, it's broken. I cannot show it. Anyway, I mean, yeah, I'm actually doing supplying different targets in different parts of the simulation, and it's working. So you can pretty confidently do that, I think, with NCAs.
That's interesting because in the embedded NCA scenario, is this the one, if I remember rightly, where the final image is like it has to make a circle of a certain color over in a certain part of the image to make not a certain color.
Just a certain part of the image. Oh yeah, it's working now. But if we initialize with a random pattern and we toggle it, oh, it's actually running really slowly now because my computer is struggling, and I'm just toggling it just to stop here. And yeah, it's so what I do is like you have the initial pattern. I run the simulation for, let's say, that many steps, and I supervise it to look like a star, just so that I say that this is, in a way, I'm exploding the information of the initial digit so that all of the cells, in a way, get informed of what was written here. And then I just supervised on the position. I just supervised like an actual image of a circle in this position. And because of the star, and the star is kind of different based on the initial image, you get depending on how different it is, it produces a circle differently on in a different place. Yeah, and you actually do
I didn't expect.
What did you say?
Oh, it's just a passing comment. I said I didn't expect that it would have a huge impact, but it's cool that it does.
Yeah. Okay. And yeah, that's that. Is anything interesting here? Let's see. Simulation rollout. Do they have anything with NCAs here? They can visualize the space. Random simulation. Substrates. Oh, here they have NCAs. This is plania. This is particle linear. Okay, this I didn't see that. They don't have it in the article actually. But I guess they have experiments here with particle linear as well, which is nice.
I don't think that's in the paper either. Or maybe it's in the supplementary.
Yeah, I mean, they also only have particle life in the paper.
It looks like there were some NCA explorations in the notebook, just below the P linear. Oh, that's random. Sorry, that's random simulations.
DNCA. I'm not sure what DNCA stands for.
Oh, actually, I don't know.
CA. So they have particle life plus, particle linear, particle life voids linear, NCAD1, NCAD2. I'm not sure if they give explanation of what each of those substrates are, but
If I had to guess what the D was, maybe deep.
I don't think there is a deep NCA. That's interesting. I haven't seen anything on deep NCAs, but I have thought about doing an NCA and making it more deep. I guess they haven't included them in the paper because they're not really that interesting and diverse, would be my guess, because looking at the results here. So what is the experiment they're doing here? Is this the...
I think this is just showing off random ones. Random examples of the different substrates.
Oh, all of them are... Oh, yeah, all of them are random. Yeah, okay. I guess they should have something more interesting here. Simulation rollout of this thing. I'm not sure if they haven't, why they haven't run, or they don't have anything on optimizations on these models here, but might be in the next paper.
I was going to say something for future work.
Yeah.
Do you have any other suggestions for what future work might be in this area, other than exploring NCAs more?
Yeah, most definitely more NCAs. I think what they give us for future work covers it well enough, like embedding temporally, like sequences. Maybe also
Do you think there'd be any way to expand their definition of open-endedness? Because they obviously focus a lot on diversity and novelty through time in this. Do you think there's any new dimensions to the space that could cause some more interesting emergence?
I think so. Yeah, I think embedding temporally could give you the ability to explain more and maybe like propagate signal in a better way to describe the actual complex behavior that you want to see. Because here you're kind of guiding it by, I want to see this specific image and this specific image and this specific image, which is a little contrived. I mean, I don't know, might not be.
And what would be interesting to me would be to see some sort of semblance of an interaction happening, like how in flow linear or similar models we get examples of things having persistence of structure, or when they bounce into each other, that sort of thing. Like imagine that would be quite hard to find in a snapshot-like fashion, like is currently being done.
Yeah.
Whereas describing a collision where the two bodies don't dissolve, if that could be effectively embedded into the embedding space and looked for, that could provide some interesting novelty.
Yeah, yeah.
Or even computation, in the sense that, like, by interacting, the state changes in some way. So, like, it changes color when it bounces into something.
Yeah,
Like,
yeah. This is the
feels
type of
quite-
diversity that I wanna see. Yeah, computation, something for them to make something. I mean, sensible in the context of the simulation itself. Like, I don't
Yeah,
know.
not expecting you to start writing English words, but like
Yeah,
something a touch
yeah.
more complicated than
Like
or
consuming resources and like fighting with each other, things like that.
yeah, that would be pretty cool. And I can
Yeah.
see how having temporal information would be quite useful for that, because it's all about the interactions at that point, not the static state which it reaches
Yeah.
after an arbitrary number of time steps.
Maybe that's why I'm kind of not sure I like this approach, because apart from you explaining it in what you want to see in pictorial form, you kind of can't really. I mean, I guess what you can do is explain. I guess they do it here, but where was it? They they explain here. They have predatory prey dynamics. That would be really cool to see, but I just see a picture being generated of things that look different from each other. Like we have like things that are kind of different species here. If you look squint your eyes closely,
Yeah.
like there are things that look like monkeys and like aliens, but they do not actually even. They don't even have behavior. They're quite static.
I think that subfigure highlights that exact problem. They're asking for dynamics in a static image.
Yeah.
that's kind of always going to be a limitation in this case.
Yeah, and I think you have to go away from the paradigm of actually supervising on what you want to see, and I'm envisioning just a different type of just optimization signal. I don't know. Maybe more, something more akin to intrinsic motivation. Something that's not that direct. I guess I'm not sure how to describe it.
No, I completely agree. I think that last bullet point there is the important part of this paper. It's human aligned representations.
Yeah,
I think that's what they were really going for in this work. IMO.
it's nice that you can search in that space that looks nice to humans. That's powerful in a way. But just the procedure itself, I think maybe this method that they have combined with something else that's not like directly related to. I guess you can still work in the space of clip where everything looks nice, but I don't know, just I'm not even sure how to
Yeah,
describe
I think this serves well as like kind of starting point, right? You just you have some rough idea and then you just give it a prompt and then give you something to work with and then you. But yeah, I agree. I think this on its own is quite simplistic. You can't really find anything that's very rich with just prompting alone.
Yeah, Piotr had a question which I didn't see, so maybe I missed the part where you were explaining the part, but is there search algorithm optimization Steering simulations to get interesting results, or more like browse through the search space and find interesting solutions?
I guess this
There was
is
part of the genetic algorithm, right?
Was that?
There was a part of this in the genetic algorithm of optimizing towards diversity.
Yeah, there's part of it in both of the open-endedness and the simulation. In the open-endedness case, you supervise on a single trajectory, and you want to make the single trajectory be very cover a lot of space, a lot of positions in clip space, very big area, and this would mean that it varies in clip space a lot. And this is how you get like open-endedness, which is a simulation that's continually producing novelty, which is nice. And here they're kind of trying to find a set of different trajectories that are very different from each other, and they're just here they're optimizing to find a set of very diverse simulations.
I have maybe a very annoying question to ask.
Yes.
Is this open-endedness, or is this just a big search space with multiple solutions that look novel?
Yeah, I was just thinking that too, actually.
What's the
I'm not sure
definition
about
of
full definitions of
open-endedness
open-ended.
that you want to that you what is your definition of open-endedness? I mean, what is actually
So yeah, so I think if you have the caveat of this being open-endedness through time, then yeah, I would agree with that being like an acceptable definition. But then, if you have boundaries, you have a search space. Well, open-endedness is what's beyond that search space in terms of how I see open-endedness. So it's like increasing the dimensionality through time as, incorporating new information.
May I butt in the discussion? I think that the classical example of open-endedness is true biological evolution. And when I mean biological evolution, I mean something that is potentially infinite. Like really infinite.
But our universe,
And I don't...
from the way we understand it, I think it's also a finite system, so I guess we can.
But you can never prestate all the possible forms that a biological system can have.
Yeah, that's. I'm not sure that's. I mean, if you look at physics.
Still, you don't know how many different organizations can a biological system have. It has a potentially infinite number of possible states, and I don't think that any computer simulation as a fact can even scratch the surface of this huge multiverse of possibilities that real life can experience.
I agree that it would be very compute heavy, and we are nowhere near trying to simulate every possible interesting thing there is to simulate. But I think I've heard like estimates of all the possible I don't know particle.
I am using the definition given by Stuart Kaufman, if you have ever read about his work. He is the one who first put forward the question of, is it statable? Are all the possible forms of life that an organism might evolve into pre-statable or not? Yeah, Stuart Kaufman. Stuart.
Sorry. Stuart.
He has several books on the topics of biological evolution.
This guy.
Yeah, this guy.
Check it out.
He has interesting books about biological evolution and its possibilities.
Great. To maybe to answer again the question of Nathan, maybe we need more complex simulations where we run more complex models where we run that, because here you say from what I understand is that because the space of the simulation is very small, you cannot really talk about open-endedness, even if you like find simulations that traverse like very big spaces, very big like wide paths in that space. You're still in very limited space compared to what biology and real life has, and in that sense it's not really open-ended. But yeah, I think from the way I see it, you just need more. I guess you with more resolution you might be able to achieve something that resembles more something that's more open-ended. I'm not sure if I.
Yeah, I mean, I knew it was an annoying question. That's why I prefaced it with it, because I think it just depends on like your, the philosophy or ideology of your research and the domain that you're part of.
Yeah.
I'm just curious. I sort of looked at open-endedness as a property of a system or a simulation or something like that. It has the capacity to be open-ended, which to me, again, depends on the definition. But to me, it makes the search for open-endedness within a system more of an attempt to prove whether or not the system can change in such a way that there'll always be more things that you could change.
I would like to butt in and say that there was one article, I can't really find it right now, by Monsieur Moreno, Spanish researchers who were studying the definition of life as a consequence of the possibility for open-ended evolution of the biological systems. And they gave a very interesting definition of open-endedness in their paper. As far as I can remember it, I'm not citing it correctly. I just want to point this out. Open-endedness is when the system acquires the ability to evolve beyond the limitations of its environment. If we can apply this definition of open-endedness, then a computer system would have an open-ended evolution if it starts to have some behaviors that cannot be prestated by what other systems usually do in such an environment. If you can understand me, or should I be more precise?
No, that makes sense. So is this a fundamental limitation of ASAL? Because if you're specifying the search space, you can never really go beyond it.
But then, if that is the case, if that is true for open-endedness, have real biological systems, when did they did something beyond their environment?
If we assume the environment was originally, say, soup, I would argue that maybe the first formations of RNA and DNA were a moment when a system within that environment.
I would like to butt in and just point out something interesting. If you just have an RNA or DNA or whatever chemical systems you have, they would strive towards a chemical equilibrium. Do you understand me? They would have an evolution path that might be very big in diversity, in size or complexity of the molecules that are being produced. However, it would tend towards some kind of a chemical equilibrium. However, if you have a truly living system, this living system will try to break the chemical equilibrium and actually self-replicate beyond the possibilities of the environment to carry it out its replication. So you can have a definition of open-endedness where you can mess with this quality of living systems to go beyond what is permissible in terms of their numbers by their environments. Do you understand the difference between a system that is replicating purely in order to produce more copies of itself and thus breaks out of a chemical equilibrium, and the system that is only trying to achieve a chemical equilibrium no matter how complex it is?
I can see the difference on paper, but I'm just trying to think of an example that pleases my mind. Taking the RNA example, so at what point did am I getting it right here that sort of life is the open-ended search by biology.
Yes,
Or
it
by
is.
biology. Yeah.
And
Okay.
I would also
That's
like to add that maybe indeed this open-endedness has some infinite component. I know that infinity is a very tricky concept for physics and for the real world at all. However, biology might just employ a sort of it because it really has an unlimited potential to innovate. And you need a search space that is also potentially infinite in order to accommodate such a system.
I think I'll still need a little bit of time to wrap my head around the idea of there being genuinely infinite possible variation. That still doesn't quite sit with me properly. But I do think that I would be happy to say the space is enormous. But in my mind, any sort of computable system, that's a statement, anything with a finite number of states is going to have a finite number of possible kind of configurations.
But if it has a finite number of possible configurations, wouldn't it just strive towards an equilibrium?
I mean, that's a good question. You could argue that the universe might be a la the second law.
I would argue that maybe there is indeed some component in the possible number of organizations that a biological system can have that is actually infinite.
Okay.
Like real infinity. However, I understand that this is going to be a matter of debate, both for computational specialists and for biologists and chemists too.
That makes sense. I like what you've written there, Andy, in the comment. In the sense that there might be a search space defined by physics and chemistry, but then there are examples of very interesting changes in the kind of complexity in the environment that are brought about by the system using those rules to change the environment, and thus themselves by evolution.
Yeah.
That's interesting. Thank you. Is your name Jordan? Thank you.
Yes.
Nice to meet you. And also, that was good.
My specialty is precisely theoretical biology and philosophy of biology and origins of life and the sort of things. The interesting big questions in biology.
Fantastic.
All right. Great stuff. I mean, if there's nothing more, I guess we can. That could be for today. Oh, actually, maybe we should discuss next month's paper or decide on it. And maybe if the discussions are over, I can
Yeah, actually,
stop the recording.
Actually, I have a suggestion for the next month. I will put it forward in the server a couple of weeks ago, or maybe 3-4 weeks ago. In the synthetic biology section of the server, I cited one